New technology in use as a research tool at Nottingham University Hospitals, NUH, has the potential to help laboratory scientists and pathologists diagnose brain tumours from the moment the neurosurgeon makes their first cut. Consultant neuropathologist Dr. Simon Paine explains how nanopore sequencing devices has been employed in research involving NUH and the University of Nottingham, and why this application can be a game-changer for patients in the future.
Nanopore is a really funky way of sequencing DNA. It's a little hole in a membrane, and the DNA goes through that hole, and as it goes through, in real time, you can detect the code of the DNA. But you can also identify the changes around the DNA. So the things like methylation groups that are stuck onto the DNA, as the DNA goes through the pore, because of the change in the electrical current and some really fantastic computational work, you can then sequence the DNA and understand those changes around the DNA in real time. We're using this technology, the Nanopore sequencers, to work out what sort of brain tumour a patient has. Currently, that process takes several weeks, and it involves us sending tissue away to London. Whereas with Nanopore, we can do this in the operating theatre suites. The block behind me is actually the neurosurgery theatres, and we've actually done it in that suite. It's a very small box that allows you to analyse the tumour very, very quickly, to gather the genetic data that you need in order to classify tumours. So as we've understood more about the biology of brain tumours, we realise that it's those molecular changes that really predict a patient's future. Those are the things that take us a long time currently to identify, but with the pathway that we've developed, we can do that within a couple of hours to get a diagnosis, and then get the remaining important molecular diagnostic data over the next day. There's enormous potential to improve patient outcome here. But I think more broadly, the pathway that we've developed could be applied to other tumour sites. So, for example, sarcoma or leukemia, lymphoma, the same sort of technology that we've been working on could be applied to those pathways too. And so potentially, this work could improve the accuracy, the speed of diagnosis for these patients too. The main aim of our project is to develop a pipeline, a pathway, a way of diagnosing brain tumours that's quick, it's efficient, and it's accurate, and that it's easily adoptable by other places. So one of the problems in the UK at the moment is that there's a real variability in access to the molecular testing that we really need for our patients for their tumours. The Nanopore sequencer, it's a small box, it's relatively cheap, it's relatively easy to use, and so it's got the potential to improve that equity of access to genetic testing for patients all over the UK. So, working as quickly as we can to get it safely into clinical practice, we're hoping that over the next few months we'll be able to get it into routine clinical use. And we're also working with colleagues elsewhere in the UK so that they can adopt the pathway that we've developed, so we can gather more data, more experience of using it, in order to get it validated, get it approved for use.